Hello, students, and welcome to my screencast on meiosis. Don't worry about cell differentiation up there. So this one's going to be a short one just to get you ready for your test. I'm going to use a lot of Khan Academy images, and those are all available on khanacademy.org. All right, so here is the basis for meiosis. And we modeled this with drawing it. We modeled it with pop beads, and then we also used straws, or tried to do this. So this would be for a cell 2n is equal to 4 chromosomes. Here you can see the 4 chromosomes. 1, 2, 3, 4. All right, and to do the cycle of life, they need to make sex cells that have 2 chromosomes in them. So that when that sperm and egg combine together, then the baby would have 4 chromosomes. And then it can undergo mitosis and make lots of copies of that cell, and then some of them will become differentiated, and you'll get a beautiful baby kid. All right, so here is a homologous pair of chromosomes. Those homologous chromosomes, homo meaning the same, have the same genes, but they may have different versions or alleles of the gene. So I told you this one might have eye color on it. This one could have a hair texture. All right, so here's homologous pair 1, and here's homologous pair 2. So what happens during prophase is they condense, and they form a tetrad. This is different than mitosis. They're going to hug each other, and as they're hugging each other, special enzymes will help to swap sections of the chromosomes. And here you can see that they swap this purple part and this pink part, right? And I had you draw that in class. And they're swapping alleles, and this happens on every set of homologous chromosomes. 1 through 22 are the autosomes. And it also happens on the sex chromosomes as well on the pseudo-autosomal region of it. So anyway, they swap these alleles here, and now they're going to get pulled apart. This is telophase 1. It uses the same names as mitosis, PMAT. And here at the end of telophase 1, once the cells split, the cells are fully haploid, right? They have half the number of chromosomes, 1, 2, 1, 2. So that's great. But they still have too much DNA because they have these sister chromatids. Now, the trick is, unlike mitosis, these sister chromatids are no longer identical due to crossing over. So here's an example of crossing over. This is what I was trying to get you to do with the straws. It didn't really work, but it's okay. All right, here is meiosis 2. So now they're going to line up. It's going to be just like mitosis, all right? And they're going to get pulled apart. Now, an interesting thing is it's completely random which way they're facing during metaphase 2 here and during metaphase 1 here. And that random alignment of homologous chromosomes provides genetic diversity as well. And so there's actually 2 to the 23rd possibilities of the ways they can line up. Here's an example of Play-Doh, and I'll try and do this with you on your review day. So this is just going to be 2n is equal to 2. Now they double up during the S phase of interphase. Here they're going to hug each other. They're going to swap some sections. They're going to get pulled apart. Haploid, right? This is one homologous pair, one homologous pair. So we went from 2n is equal to 2 to now n is equal to 1. And now they're going to get pulled apart, and you have 4 sex cells. Right? These are called gametes. In females, 3 of these will die as polar bodies, and they'll contribute all their nutrients to one of them to make a bigger egg. In males, all 4 of these will become sperm. So here's a little animated chart for you for meiosis, and you can see them making genetically diverse chromosomes. So here it goes 46, 23, and 23. All right, I'm going to post an old video I made about all those terminologies, homologous, haploid, diploid, etc. And keep studying.